In biomolecules, and our lesson today is over nucleic acids. So we've already done carbohydrates, we already did lipids, and now we're in nucleic acids. Now this is probably the most important one for you guys to understand because this is going to be your DNA and your RNA and your genes and a lot of other stuff that's related to the more important topics in biology. And what you're going to do today is actually a multi-part assignment. You're going to do a virtual DNA escape room and also take your C notes for nucleic acids. So as you go through this escape room, there'll be 3 videos, and then after the video, you'll complete portions of the escape room. Well, just like any other video, while you're watching the video, I want you to take C notes, so handwrite your C notes, and then use that information to answer the questions in the escape room, and then type up your notes once you're done with all of that into your notebook. Our essential question for today is, what is DNA and why is it important? Now, once again, we are in our 3rd part of biomolecules, which is nucleic acids. Remember, our biomolecules are our carbohydrates, our lipids, our nucleic acids, and our proteins. Our carbs are sugars, our lipids are fat. Our nucleic acids are pretty simple. It's DNA and RNA. And then the next one we'll do is protein. Make sure you know what elements are found in each one of those biomolecules. So we always been doing carb, lipid, acid, protein, cho, chomp, chon. So since we're doing nucleic acids today, you know that the 5 elements that are part of nucleic acids are going to be carbon, hydrogen, oxygen, nitrogen, and phosphorus. All right. So our first key point is simply, what is DNA? You guys already know this. DNA is really called deoxyribonucleic acid, and it is that blueprint for life. It is the instructions for building proteins which kind of regulate and control life. So you already know this. DNA makes RNA and RNA makes protein, and that's really all DNA does. You know, your genes are on your DNA, and each gene is going to code for a single protein. Now you do need to understand the structure of DNA or what it looks like. So the shape of DNA, or that structure of DNA, is called a double helix. And the reason it's called a double helix is because it's double-sided. So if you look at my picture here of this big long strand of DNA, and here it's kind of zoomed in a little bit, and then it's really zoomed in here. But DNA is a polymer. It's a very, very long polymer, which means it's made up of a whole bunch of monomers. And if you look really closely in this picture, you can see there's some monomers in here, all these different colored things. So my monomer for DNA is called a nucleotide, and this is one of them right here. This is a nucleotide. Here's a nucleotide. Here's a nucleotide. And then on the other side, because it's double-sided, here's another nucleotide and another nucleotide and another nucleotide. And these nucleotides, there's thousands of them, come together in this structure, and then it twists. And that's why we call it a double helix. Double because it's 2-sided, and helix because it's twisted. I like to refer to it as a ladder. You also hear it referred to as kind of like a zipper on your sweater or something like that. But it's like a ladder. So here's my rungs of the ladder, or here's my sides of the ladder, and then the colors are going to be the rungs of that ladder. Now, to really understand what DNA looks like and how it's made, you need to understand what a nucleotide is. So a nucleotide is the monomer for DNA and for RNA, and this is really important for you guys to understand. So I'd actually draw one of these in your notes. There are 3 parts to this nucleotide, and you'll hear me refer to them as a house, a pool, and a basement. And the reason I call it a house, this right here is going to be your house, is because it is a pentose sugar, and that word pentose reminds me of the word penthouse. So pentose sugar just means it's a 5-carbon sugar, And there's two different types really that we're going to see in our nucleotides. One is called deoxyribose. The other one is just called ribose. Um, Deoxyribose is a pentose or 5-carbon sugar that's found in DNA. And then ribose is going to be a 5-carbon pentose sugar found in RNA. So here's my house. It's a pentose sugar. And here's where my carbon, my hydrogen, and my oxygen come into play, because sugars are carbohydrates and carbohydrates have carbon, hydrogen, oxygen. So right in here I have carbon, hydrogen, oxygen. And remember, in DNA and RNA I also have nitrogen and phosphorus. Well, that nitrogen comes right here in this nitrogenous base, and the phosphorus comes up here in that phosphate group. But going back to the three parts, we got our house, our pentose sugar, then we have a swimming pool, and I call it a swimming pool because of the letter P. That P is for phosphorus or phosphate. So I have a house with a pool behind it, and then I have a basement. And out of these three parts, the only thing that ever changes is going to be the nitrogenous base. In fact, every living thing, no matter what it is, a plant, a tree, bacteria, you have the exact same sugar and the exact same phosphate group in that order. So it would go sugar, phosphate, sugar, phosphate, sugar, phosphate. And that right there is kind of the side of the ladder. And this is called the backbone of DNA. And then the nitrogenous base is what actually gives you your traits and makes you different than other living things. And it's what makes you different from each other. And it's the order of these bases that make us different. So here's my nucleotide again. So I have a house, I have a pool and a basement, or a sugar, my phosphate group, and my nitrogenous base. And the important thing about these nucleotides is going to be this base, or this nitrogenous base. And there are 4 possible bases. The 4 bases are adenine, thymine, cytosine, and guanine. And we're going to just abbreviate them with the first letter of each word. So A for adenine, T for thymine, C for cytosine, G for guanine. Okay, so you could have one of 4 different bases here, A, T, C, G. Now, what makes you different than other living things is going to be the order of these bases. So, this order here, A, T, C, G, that might mean you have brown hair. Well, if I change this order, T, T, C, G, maybe it means I have red hair. So the order of these bases are going to be what's different between all living things. Now, these nucleotides are going to come together to form the polymer, which is going to be DNA or RNA. And this right here is DNA, and the reason I know it's DNA is because it is double-stranded. So here's one strand, and here's the other strand, and they're both made of nucleotides. So here's one nucleotide. I have a house, I have a pool, and I have a basement, which is an A. Here's another nucleotide, house, pool, basement, that's a C, house, pool, basement A, house, pool, basement C. Then on the other side, I still have house, pool, basement with a G, house, pool, basement with a T, house, pool, basement. And I think you guys get the picture here. But when I add these nucleotides together, I'm going to form DNA. And one thing you might notice is in DNA, they go in different directions, right? So my nucleotides are going one direction on this side of my DNA, and on the other side, they're going a different direction. Now, these nucleotides base pair together, and this is really important for you guys to understand. It's called Chargaff's rule. It's named after a scientist who figured this out. But if there is an adenine, an A, it will always pair with a thymine or a T. So A always pairs with T, or what I always like to say is apples grow on trees. And then if I have a cytosine, it will always pair with a guanine, or C always pairs with G, or my car will get parked in the garage. And you can see here I had an A on one side. Well, it's going to base pair with the T on the other side, and C will base pair with a G, A to T, C to G, and so on and so on. All right. One other important thing about the structure is going to be how these two sides stay together. So here's my nucleotide. Here's a house, here's a pool, here's a basement, here's a house, here's a pool, here's a basement. And now this happens to be an A, it's going to pair with a T. If this happens to be a C, it'll pair with a G, so on and so on. But the way these things stay together, that A and T stay together, is through something called a hydrogen bond. And you might see that on your STAR test. That's why I'm telling you about it. And A to T has a double bond, where C to G has a triple bond. And that's kind of interesting because we've learned through research that a lot of our DNA mutations or changes in our DNA happen with adenines and thymines. And one of the reasons might be is because it has a double bond instead of a triple bond. So that C to G bond maybe is a little bit stronger than that A to T bond, which allows those things to have more mutations. All right, so with base pairing, it's actually kind of fun, and it's pretty easy once you get the hang of it. As long as you know these rules, A pairs with T and C pairs with G, you could take one strand of DNA and figure out what the other strand of DNA is. So here's my strand of DNA, A, T, T, A, C, G, C, A, G. So this might be one gene, it makes one protein. Well, it has a corresponding side, right? So that A will always pair with a T, that T will always pair with an A, and eventually it'll look like that. So here's my double-stranded DNA, A to T, T to A, C to G, G to C. And they always pair together. And this is called Chargaff's rules. And you guys actually might have to do some math based on this. So the number of A's always has to equal the number of T's since they pair together. And the number of C's always have to pair with the number of G's. And you guys actually might get some question like on your STAR test where it says this organism has 30% adenine. How much cytosine is in that DNA? And it's pretty simple math, right? If it's 30% A, that means it's 30% T, and 30 is 60%. That means I have 40% left over because 100 minus 60 would be 40. So between my cytosines and my guanines, that has to equal 40%, and it's half and half, right? So 20% is C, 20% is G, and we'll probably do some more math, get you more practice on that. But that's Chargaff's rule. All right. Next key point is RNA structure. So we looked at DNA. Remember, DNA makes RNA, but it looks a little bit different. So you guys already know DNA makes RNA, RNA makes protein. Here's the real word for RNA. It's ribonucleic acid, and this is the code for a protein. So looking at this picture, you should notice that it is not double-stranded, right? I still have nucleotides, house, pool, and a basement, but I don't have a double strand of it. It's just a single strand. So DNA makes RNA, and that's called transcription. We'll talk a little bit about this later. Then RNA makes protein, that's called translation, and we'll talk about that later. So RNA is also made out of nucleotides. There is one little difference, actually two little differences. The sugar in RNA is not a deoxyribose. It is a ribose sugar. It's still a 5-carbon sugar, still a pentose sugar. And then there's no T. So I still have my bases. I have adenine, uracil, cytosine, and guanine, or A, U, C, and G, instead of A, T, C, and G. It's also single-stranded, and it translates the genetic code of DNA into the amino acid sequence of a protein. That's a really important sentence for you guys to understand. And we'll talk about translation a little bit in a minute. So if I have a strand of DNA that's got a code for RNA, I could figure out what that RNA looks like using base pairing, right? Now remember, there's no T in RNA. Instead of a T, it's going to be a U. So in DNA, you know, that A would pair with a T, but since there's no T in DNA, or in RNA, it's going to pair with a U. So it would go A to U, and then everything else will be the same. T to A, T to A to U, C to G, G to C, and it should look like this. So my DNA was ATTACG, CAG. Well, A pairs with U, that makes it RNA. T pairs with A, T to A to U, C to G. So the only difference in that base pairing is instead of a T, I'm going to have a U. And that's one way you could tell a difference between RNA and DNA. So if you're looking at a sequence of letters kind of like this and it has a T in it, it's going to be DNA. And if it has a U in it, it's going to be RNA. And you know, if we give you a strand of RNA, you should be able to figure out what the DNA looks like just by going backwards. You know, U pairs with A pairs with T, A pairs with T, U pairs with A, G pairs with C. So base pairing is the same, except RNA has a U instead of a T. Here's an overview of those differences between DNA and RNA. DNA is double-stranded, while RNA is single-stranded. DNA has a T, RNA has a U, DNA has a deoxyribose sugar, and RNA has a ribose sugar. All right, so that's the end of the first video. Use that information to complete level 1 of the escape room.